A team led by Liu from China Agricultural University recently published a groundbreaking study in plant communications. They identified a key factor MSHOT that can promote corn to accept genetic modification. It does not participate in the early stages of infection, but instead focuses on the later stages, strongly promoting the formation of embryonic callus tissue. This is akin to providing corn with a regeneration accelerator. The research team selected 2 representative, difficult-to-transform varieties pH for CV, ZENG58 for verification. The once stubborn ZENG58 corn saw its transformation efficiency jump from 0% to an impressive 7.2%. Another tough variety, pH for CV, also experienced a more than 10-fold increase in efficiency. This means the door to corn genetic editing is finally open. This groundbreaking discovery is supported by the imaging capabilities of MSHOT MZX100 Fluorescent Microscope. Its 10:1 ultra-high magnification ratio and high-definition fluorescence imaging helped the team clearly capture the dynamic expression of RVP in corn callus tissue. This made the regeneration acceleration process clearly visible and the data measurable. Follow us and explore the microscopic world with MSHOT.